Welcome, everyone. This is Mark Anderson, engineering consultant with Staties Incorporated. I'm here to talk to you about exploiting statistical experiment design to accelerate pharmaceutical R&D. This is a new interface that I've only just seen in about 5 minutes ago, so bear with me here. To begin with, it'll be helpful if you would press that raise hand button or some kind of reaction button that you should see at the bottom of your screen. All right, Wolfgang, Juan, Chris. Okay, fantastic. Okay, so you can go ahead then and lower your hands, or I can do it for you. This gives me some very helpful feedback that this new interface is working just fine. So again, I thank you for participating in this presentation. And so during the presentation, I'm going to ask you to stay on mute throughout, and there may be some controls here for me to take you off mute, and there may not be. I haven't quite figured this out. So I'm doing an experiment, unplanned, along the way. And since I've used a lot of these different video conference interfaces on Teams and Slack and so forth and so on, I think I can get it done. But it'll be just a little extra challenge for me. By the way, if you do have questions, since I may or may not be able to pick them up during the presentation or afterwards, your best bet would be to send questions to this email that I'm going to show you. So let's see if this works, if I can write on the screen with my little tablet here like I've done in the past. I think it's going to work. So this is what you want to make a note of. Mark, that's my name, at staties.com. And send questions, that you may be jotting down throughout the presentation directly to me, and I'll get right back to you on that. That would be very helpful because it'll help me improve my presentation as I go along, seeing where maybe people got thrown off a little bit. Or you may question, some things that I assert in the presentation. I'm very happy to be challenged by that as well. Or it could be there's some things that I don't talk about here that are confronting you, and I'd be very happy to help, break through for your own experimentation. Hopefully, the recording of this will be posted to our YouTube channel and also the slides, and you can get to that through this link, 3wstaties.com. Go to the webinars part of it and follow through, and you'll be able to find the presentation and then track it, back to the YouTube presentation if you want to watch the video. So just to get some exercise here, press that raise hand one more time if you're with me so far on slide number 2. Okay. And Chris, thank you for weighing in and all the rest. All right. All right. So what's in it for me is, of course, a big question to come into a presentation like this, or looking at the recording on YouTube at a later date. And so here's what I was thinking, the value of this presentation would be that you would learn how multi-component and multifactor design of experiments, and we'll use the acronym DOE for that, tools will empower you to quickly converge on a quality by design sweet spot, which are ingredient and factor settings that meet all specifications at minimal cost. Now, this idea of quality by design is something more specific to pharmaceutical and other regulated industries. It's a really cool idea that if you can find a setup for your formulation and or process that consistently meets all the requirements, then certainly this would be a great thing as far as the FDA is concerned, or the European regulatory authorities. So I like the idea. I don't think it should be just restricted to regulated industries, but I think it's something that all industries can make use of. My background being specialty chemical, and I did a lot of work in R&D, developing processes, scaling them up, bringing them down to the plant environment, and then wrestling with the people in manufacturing to try to get this into that sweet spot and keep it there. If you can develop a process that works really well, but also is robust to environmental conditions and such, variations in the inputs themselves, etc., then you really do have something that hits the spot. So that would be, probably the main take home. But I think really all engineers, chemists, scientists that are working on development of drugs, pharmaceuticals, will do well by attending this, and specifically learning how to do process screening to detect previously unknown vital few factors, mixing those together with what is affecting your process, and then doing an in-depth characterization to detect interactions, which are oftentimes the key. And then if you detect that there's nonlinearity through curvature testing and such, you may want to move on to the pinnacle of DOE, which is response surface methods, or RSM. Now, all of these approaches of screening, characterization, optimization, which come in through RSM, translate over to formulation development, but using a specialized toolkit called mixture design. And this would be, very important for developing active pharmaceutical ingredients. On the processing side, I think that would be more of manufacturing of generic pharmaceuticals, but it certainly applies to manufacturing of APIs as well. So hopefully, whether you're a newbie to design of experiments or you're experienced at it, and I recognize some of the names in the group are very experienced at doing DOE, this talk will be helpful to you and your organization's bottom line. I've found that over the years, when I've had what I recognize as some very advanced practitioners, when I looked over the list of people raising their hand, and I wonder, since I'm presenting this more at a basic level, what the values are. But I learned early on one time when I was talking to a group in Detroit after publishing my first book, DOE Simplified, and going on a little road trip to promote that book. I had a PhD statistician come in from Kellogg in Battle Creek, Michigan. And early on, I sussed it out. I said, what's your background? Do you know what a p-value is? And most people raise their hands. Have you ever done a two-level factorial design? And very few people raise their hands. And I just kept escalating, and I couldn't get to the bottom of it with this one individual. It turns out that she was a PhD statistician, as I mentioned. So I said, why are you attending this presentation? She said, well, I really want to take the tools of DOE and deploy them throughout my group of researchers, so they're not always coming to me and making me do the design of experiments and then coming back with their data and making me do the analysis. I would like to get it set up with the proper tools, which Statis provides for software, and some know-how so that they can do most of that themselves. And that's really the philosophy that Statis has, which is not to do the fishing for you, but teach you how to fish. And so that's one of the main goals of this presentation, is it giving you some ideas of what DOE is all about, getting you interested in that, and then having you develop your knowledge of that and put it into practice. And then for those that are expert, hopefully you can pick up a few ideas from me on how to make that same pitch to people in your industry or within your company. So what I'd like to do to begin with is really take a high level view of what's going on here and consider that we have some kind of a process or a product, or even just we could call it a system. If I was talking to people in medical device industry, for example, I might use that word. But I think for the pharmaceutical industry, we're primarily talking about process, but we could be talking about product. So what you do is you need to think about is from a series of what, as a chemical engineer, I would call unit operations or unit ops. Which one of those steps is the one that is limiting to you in terms of the yield, in terms of loss of quality for purity and things like that? And for example, I'll just tell you what I worked on early on when I worked with General Mills Chemical, which became part of Henkel, and it was a natural vitamin E product. And this natural vitamin E was sold to pharmaceutical companies, who then did the tableting and packaging and things like that. But we would produce, something that was 99% pure alpha tocopherol, which was a natural form of vitamin E. So that was one of the goals that we had, was the purity. But what was really important was the yield of this natural vitamin E, because it was lost quite a lot along the way because of it being an antioxidant. It was, fairly fragile coming from the raw material, which was soybean oil, deodorized or distillate, to the final pure product. Their yield was quite a bit lower than what it should have been in theory. So they tasked me with doing some research and development in Minneapolis and then going to the plant in Kankakee, south of Chicago. And the first thing I did is I said, well, look at the overall process, which part of it is where the yield is being lost. And it turned out to be in a unit operation, which was the crystallizer. It was at the very end of the whole series of unit operations. Now, what you do once you develop a focus on your process or product is then apply first principles with your expert, practitioners in terms of, let's say in this case, of course, heat transfer would be a key aspect. If you were doing a chemical reaction, you might be looking for some input from PhD organic chemists and such on the kinetics and things like that. So first principles come into play. Another thing to consider is actual operators, people who are running that plant. They may know some things. So with that in hand, you can apply some kind of a brainstorm. I like the fishbone diagram, and in this case, our focus at the head of the fishbone was the yield of the product, and then we consider, what materials are coming into play, what kind of machinery do we have, what kind of process or method do we have, you know, is this impacted by the people who are operating this? Yes. And then what about environment? Yes, that could be very important. And you come up with a brainstorm, and from that brainstorm, we then develop an idea that we're going to do a series of tests and make purposeful changes to certain input factors which come out of this brainstorm. And so we develop a plan, and we might, for example, if we consider the letter K to be the number of factors, we might start off with, let's say, 8 factors at the most, and possibly do a screening. And I'll talk to you about the strategy as we go along. But these would be deliberately controlled. We don't just let the process run, which is what the manufacturing people wanted me to do. They said, go ahead and do all the experiments you want, but just don't change anything. Ha ha. So that wasn't going to work. What we want to do to really understand cause and effect is make deliberate changes in the input. And then, of course, we're going to look at what happens to the outcome in terms of the responses. And these are the Y's. This would be Y sub 1 and Y sub 2, and we might have a lot of other responses. The inputs are the X's. And what we're going to do then is we're going to apply statistical rigor to only react to things that have a p-value less than 0.05, would be my suggestion. And hopefully we'll see that, and then we'll be able to derive a model that predicts the response as a function of X. And this is the big payoff of DOE, is that you develop these models and you can develop them for each of the individual responses, play them off against each other, and like I say, eventually get to that sweet spot, which is what I promote to all industries, but in the pharmaceutical industry, we might tack on QBD, quality by design, a space of experimental inputs that meets all our requirements and even is conservative enough that we're fairly certain that it will be reliable. Okay, so I hope you're thinking about that for your own process, that something's in your head right now. Yes, raise the hands if you're still with me and you're thinking about something that you might be working on for your company or for your own, fun and education, like, mixing up a nice little wine cooler here this summer could be an experiment. Okay, so Ruth and a bunch of other people are weighing in, Marcus, etc. Thank you. Okay, so now for those of you that are new to design of experiments and are pretty much, set up doing the traditional scientific method, which is hold everything constant and change only one factor at a time. I call it OFAD. You need to know that a multi-factor strategy is going to be much more effective. It's going to allow you to develop things a lot faster. And I did some research recently. In fact, this just happened earlier this week, and I found some statistics. DOE led to 40% experimental time saving. Design of experiments halves the development time. Industry benchmarks indicate that you can get a 50 to 70% savings in time and cost. And I think this is probably the biggest pitch for the research managers, and the people that are sponsoring your experiments. But there's a lot more to it. But the main thing is that by applying a parallel processing scheme rather than one factor at a time, we can end up doing things a lot faster. For example, here's a real life experiment. It was a life study from an accelerated test for the shelf life of a pharmaceutical. And it turned out that they were having some issues with this pharmaceutical. It wasn't giving enough life, and they needed to actually double the life. So their goal was to get twice as much life as before. With this accelerated test, they could test it fairly quickly. And currently, they're running at 17 hours, let's say, in this highly accelerated test. They need to get it up to 34 hours. So you get your brain trust together, and these engineers and scientists thought about this, and chemists, and they decided that factor A would be the most obvious thing to increase from its current level up to a higher level. And notice I've got this coding of minus and plus, which is a standardized coding for levels of things that are numeric going from low to high. It could be also a categoric factor. So one way or the other, it's changing from one level, our current level, up to a new level. But notice that the result wasn't very good with 25 hours. So that wasn't satisfactory. So they put their heads together again, and they thought, well, let's try factor B. That would be our next best guess. Let's try increasing that. Well, it worked a little bit better, but maybe not significantly so. But certainly it fell far short of that goal of doubling. Nope, that's not going to work. So then they had one other idea. They said, well, we don't think this is going to work, but let's try changing factor C from low to high. They were pretty sure it would actually make things worse, and they were actually pleasantly surprised that it didn't make things worse. And later, as kind of a postscript of the story, it turned out that the higher level of C was a cheaper condition that didn't degrade the results. And so they did end up actually changing to the high level of C. But that's not part of this initial story. The initial story is pretty obvious if you're staring at this whole time and thinking, Mark, just look up. Oh, yeah, if we would just do both A and B together in a test, we would immediately see that there's a breakthrough improvement. And this is what excites me the most. Of course, I like to be able to reduce the experimental time by 40% or 50% or 70%. I'd love to do that, but I'm really more focused on trying to have a success with my project. And so this strategy of doing multifactor greatly appeals to me as a chemical engineer because, of course, in the chemical industry, there's always things, combining. I call it chemistry. There's chemistry. You put two things together, you get more than you would expect from either one alone. And this would be a synergism. And specifically, it's an A-B two-factor interaction. And only by doing multi-factor do we detect that, right? So why would you ever just do one factor at a time? I don't get it. Other than initial range finding experiments, and sometimes as you go along, you stop for a minute, try a few things, refocus your design of experiments, and that's perfectly fine. But for the most part, when you're getting anywhere near to where you want to be, you want to deploy this multi-factor approach. Now I'm going to erase all these scribbles and I'm going to tell you more about the efficiency of the DOE, but we need to consider it in context of the power of the experiment. Of course, it's a bad approach to do just one of each. And I don't think it takes a lot of convincing, even for one factor at a time, people, that that's the case, that we really ought to do more than one. I generally recommend a sample size of at least 4 and possibly a lot more, depending on how small of a difference you're trying to detect and how much variation you're having to deal with in your process and test. But 4 would be the minimum. Now, this experiment that I've demonstrated here is actually a 2-level experiment with 3 factors. And we use this notation to indicate there's going to be 8 what we call runs in this experiment. Okay, and you can see the 8 runs listed out here geometrically. Now consider that with these 8 runs, we actually have 4 at each of the high levels. For example, we have 4 at the A plus level, which is the right side. We just combine all these 4 results, average them out. We've got an average of 4. That's got some power to it. Then we can contrast that to the 4 at the left side, which are the low A, and that also has 4. So now we've done a 4 by 4 comparison. But also at the same time, we have 4 at the high level of B and 4 at the low level of B. So we're also getting the main effect of B based on a 4 by 4, and we've got C. Now it turns out it would be too much trouble to wave my through this whole thing and show you all the other 4 by 4 comparisons, but take it from me, we also have 4 by 4 power for the AB interaction. We can detect 4 by 4 power on AC and BC and even ABC. Now the ABC interaction probably will never show up because it's a lot of things combining together, and that's part of a way that we'll consider analyzing the results at the end when I demonstrate. But on the other hand, OFAT, if they want to get that 4 by 4, they're going to have to do 4 at the baseline and then 4 at each of the high levels. So now they're going to end up with twice as many runs. So this is just another viewpoint of efficiency based on relative power. So as a whole, then you win on both sides of the efficiency, the time savings, cost savings, et cetera, and also the effectiveness, which comes out with detection of interactions of effects. Okay, what do you think? Does that help? Does that make you more excited about doing a multi-factor approach? Yes? Hands? You can see I'm a very needy person, so continue on or I'll have to stop. Thank you for that. Thank you for the encouragement. Okay. Well, there's no doubt in my mind whatsoever. I've been doing this since I started in 1974 as a summer engineer with General Mills Chemical and first learned about this approach. And I got hooked immediately. And then I had to spend a lot of time learning all the ins and outs of design of experiments. So that was quite an uphill battle, and I'm still learning. So, by the way, I have no formal training in statistics, but in the end, I've ended up writing several books on the topic. Maybe that's better because I have a real appreciation for people that don't have that statistics background but really need to get their experiments done. So, one of the early things that I worked out with a lot of help from my colleague Pat Whitcomb, a fellow chemical engineer that I started working with back in '74 and was with him when the company Statis started in 1982, and I'm still with Statis. With Pat's help, I began to understand strategy of experimentation. He had to yell at me a bit and write some things on a napkin. When he wrote it on the napkin as a flowchart, that's when I finally figured it out. This strategy of experimentation began in the 1950s with DuPont, and they called it the SCO strategy. And it stands for screening, which is to do a broad and shallow experiment first using a two-level approach. And we're going to use a fractional two-level at this point. And one experiment I would suggest that's actually a minimum run two-level experiment. It involves 8 factors, which I suggested might be a good start, in only 16 runs. I think 16 runs is better than 8. Then you have 8 at the high and 8 at the low. And you could screen down, let's say 8 factors down to maybe 2 that would turn out to really have an impact, and 6 would not have an impact. And this would be main effect screening. Now over here, we're temporarily setting aside factors we know are going to affect things. Let's say that we've got 3 factors here. We merge those together, and now at this point we have 5 factors, and this makes a good minimum run design that has a higher resolution, whereas this would be a medium resolution. And those of you who know about resolution, this is a resolution 4 design that I would suggest. So at this point, we're going to go to a high res, which is resolution 5, that'll detect 2-factor interactions. And so this would be a follow-up to the initial study, and by breaking it up into parts like this, you have a much more flexible approach for your process development. Now, what we do at this point is we add center points. These are mid-level values that literally at the middle of the square of the cube. And you replicate those a number of times. And from that, we can detect using a test curvature if it's less than 0.05, significant, yes, and it's important. Then we're going to deploy the last part of the strategy, which is response surface methods, or RSM. And these are designs that are going to have at least 3 levels. Otherwise, we skip this and we go down and we do a ruggedness test. And this actually is another 2-level design that you can apply. You can either use a Plackett-Berman, which are designs that are specialized with 4 run numbers of 4, or you can use a 2 to the k minus p, which is a shorthand for a fractional 2-level. But I would do this as a resolution 3 design. You're basically just kind of kicking the TV to make sure it still works. Yes, celebration. So let's take a look then at the beginning part of this, which is screening and characterization. And for that, I have a case study that comes out of the generic pharmaceutical. And the idea then with this experiment is to do a quick sifting and possibly even a more in-depth characterization, but using the 2-level designs to save time. This one is, this case study is a wet granulation. It uses a liquid binder to lightly agglomerate the powder, but there's an issue. It can easily agglomerate. And also you can get too hard or too soft, more friable, type products. So they've got a number of issues with this process that they have to control. So to actually put this together can be very easy with whatever your tool is. It could be Stati software. I think so. I would recommend that. But it could be another package. It could be Minitab. It could be Jump. And there's actually a number of other packages out there. In Europe, there's a package called Mode that's popular, and so forth and so on. So it's not hard to find software that has tools for DOE, but there's actually fewer that are dedicated to DOE, like the Stati software. But in any case, basically you brainstorm, you come up with some factors that you think might be affecting your process. We have the units of measure dictated here. The type of factor is, in this case, all numeric. They're numbers. But you could also have a category factor, which would be either this material or that supplier or this supplier, that type of thing. So it can be 2 types of things. Then we go to some kind of a builder. This is a builder that comes out of one of the Statis packages. But you might see similar builders, let's say in Minitab, where you have the full factorials along the diagonal. These are, in our software, color-coded white. To make it easy for our experimenters, when they go to the fractions, we indicate high resolution as green, go ahead. And then if it's a medium resolution, which is good for screening, we make it yellow. So here's one that I like, which is for screening 8 factors down in 16 runs. I also like this 5 and 16 option. Now this one happened to be 7 factors, and they didn't want to do all 128, but they still wanted to maintain it as high resolution, so they did the choice of 64 runs. That still was too many. So then what? No worries. Our software has a modern alternative called a minimum run characterization that requires only 30 runs. That's shown on this table here, and I'm going to demonstrate that for you. So we do have basically an optimal design structure that's been canned or templated for you, so you can get off the standard 2-level design builder if you really don't want to do that many runs. And if you have still enough power with 30 runs, I would recommend you consider going to the minimum run resolution 5. And by the way, we also have minimum run screening options, which are shown here. And if you're interested, go to the slide deck and you could follow up with these links to see some information, presented by our advisor at the time, Gary Ollert, my colleague going way back, Pat Whitcomb, wrote up a talk for Fall Technical Conference, which won an award. And then also I gave a talk at American Society of Quality. So now let's get to the software. Now, the first thing I'm going to ask is if you would raise your hand if on your screen you see States 360. Ava and others. Okay, perfect. States 360 is a more advanced version of Design Expert. It incorporates all of the Design Expert features with the addition of some things that probably most people wouldn't be using, which is a Python script editor. But every once in a while, we run across people that are adept at Python scripting. That opens up the whole world of Python to tie together with the States software. So that's something that comes in States 360. The other thing that comes in to States 360 again is not something that every research and development person would use. These are space filling designs like Latin hypercube, which are meant for deterministic simulations where you're trying to tease out a transfer function. And so this was something, for example, I had a class earlier this winter with the US Space Force, and they're not actually doing hands-on experiments. They've got simulations. They actually have random elements incorporated into it, but they're even doing an experiment against that simulation. And this would be a nice option, the Latin hypercube or an optimal distance space filling design. But for the most part, people can get by with Design Expert. That's going to have this regular two-level design builder. You have the nice color coding, which is why I like this. I would start here. Consider how many factors you want to experiment, and then consider at least doing the half fraction or less of a fraction that still remains green. That way you can characterize the two-factor interactions. Normally, though, if I was doing 7 factors, I'd probably say go all the way back and do a screening experiment first. Then with fewer factors, you have more options. But this particular study, they wanted to do a characterization from the start. So given that, then we shift over to this minimum run option, which has got 30 runs. Okay, so that gives you an idea of what we're all about. And so here's the wet granulation data that's been collected. And you see that we've got hardness and friability, and these are going to be traded off. And now what I would do first is I would just look at the data. And I've got a whole process here which involves first eyeballing the data, which I call an intraocular test, just to make it sound fancy, getting a sense of whether the range of it is more than we would have expected by chance, and if it encompasses some results that we like. So we're going to just kind of get a sense for our numbers here. Then I would do simple scatter plots that give us an idea of the main effects that say on the hardness of the binder granulation. And I like this little color-coded grid because it gives you immediately, when you look at it, an idea of what factors, are going to stand out with the stronger colors, either blue if it's a negative correlation or red if it's a positive. And of course, if we plot any one thing against itself, it's always, 100% positive or a correlation of 1. The other part of this that's interesting is that with this minimum run, we lose the orthogonality, the total independence of the inputs. So there is a little bit of a price to pay to go to the minimum run from the standard orthogonal matrices. But without, spending a lot of time on it here for our presentation, I just want to show you that we've got a very good way of graphically selecting effects. And it shows up in 2 places. It shows up on the left with this half normal plot, which is a plot that has as an x-axis the effect in absolute value, and we're aiming at what has the biggest effect. So we pick the biggest effect, which is a compression on the hardness, and we should be able to go back and see on the custom graphs, that would be one that is having a big effect. And so then let's go back to that. I'm using a beta version of the software, so this is what's likely to occur from time to time when I'm living dangerously at the ragged edge. So the binder solution stands out, and I won't go back to the custom graphs to show you, but if you were to go back there, you probably would see it has a big, impact. Now, as we go through this, let me clear this again. We'll see that we pick the biggest one, and you see on the right side that it starts to jump up on this ordered bar chart called a Pareto chart. But we're not done yet because we want to keep picking. Now, these are sliding a little bit because of it not being orthogonal design, which is an interesting aspect of these minimum run designs. And one of the reasons, if you go with a standard orthogonal two-level design, which would be 64 runs in this case, you would have some advantage from that of it being more powerful and also being orthogonal. However, it's probably a good savings to go from 64 to 30 and deal with a little bit of slipperiness on the effect calculations, because you can see that there's a family of effects off to the right here that tower above even the rigorous Bonferroni corrected t-value limit. And probably the only one that I would say is a little bit iffy is this AB, but I would pick it and take a look at the interaction graph and consider if that makes sense, and then possibly consider trying to screen that out later. So being a little bit aggressive on picking effects, even if they're above this black level, I'd go with that. Now, in the interest of time, I'm not going to get into too much detail here, but we're going to create a model, and this model, will maintain, certain structures we call hierarchy and things like that. We've got that built into the software. And so what we're going to do is focus on the p-values and make sure that other than terms that might be needed to maintain the structure, that they're all significant at 0.05. But most important is the overall model. And then look at the R-squared values, especially R adjusted and predicted. These are very good. Check the diagnostics to make sure that it's normal by the normal plot of residuals following on a straight line. We call it, for some way of making it more objective, the pencil test. If the pencil covers up the points like it would in this case, I would consider that normal. Watch for outliers and residual versus run, and then check and see if there's any indication that a transformation would be helpful. You can see this on something called a Box-Cox plot. You can see why it's taken me several decades to learn all this, because there's a lot involved once you start getting under the covers of this, but it's worth it. Okay, then we look at the model graphs and we begin to get some idea, is the binder solution effect on hardness, modified by granulation time? It's not a really compelling difference on the lines here. If they're parallel, it would mean is there's no interaction. It's non-parallel. So there is some sort of interaction there, but it's not super exciting. Let's look at this FG interaction. This one's a little bit more interesting. It shows compression effect that's modified by press speed. When press speed is low, which is this black line, we really don't see an effect. But when the press speed is high, we see an effect. So we work through all this. We do the same thing for the friability. And we get a model out of that, make sure that it's significant with good fit statistics, nothing abnormal. These are the three that I'd like to look at: the normal plot, residual versus run, and Box-Cox. Everything's fine there. Then we study this. There's a more compelling interaction here on friability for the binder solution to granulation. Then the payoff. Now that these models are developed, we can do a multiple response optimization, and we can simultaneously maximize the hardness. I'm going to put a stretch target on the top of this. But we want it to be at least 10, and we want it to be as high as possible. Once it gets above 16, let's say, it doesn't really matter anymore. So this is a ramp that goes from 0 and starts going up from 0 to 1, from 10 to 16. And for the friability, it's going the opposite way. And let's say that we wouldn't mind, stretching it all the way down to 0, even though that's probably not possible. But once it goes up to 0.5, it's not of any use anymore, so it's got a 0 desirability. And lo and behold, after we do a hill climbing search with desirability as a function for utility, we can come up with a solution that says the binder should be low, the granulation high, where these red dots are located. Factor D doesn't affect either of the responses, and so it's got just a flat line here. And over here, you see it's got a little cross by it that indicates it's not in either model. Okay, and then last but not least, we would look for that sweet spot that goes at least above 10 in hardness and never goes above 0.5 for friability. And so this would be a picture of it. We can also impose in confidence intervals to make it more conservative, this is what I recommend for the QBD. Okay, so that's our first example of doing a two-level design. And just to kind of check in here, how do you feel about that? Is that something that looks, valuable to some of the things that you might be doing? Okay, and immediately people are putting their hands up. I think the two-level designs are going to be the most used DOE approach, and really is, meriting us spending the most time on this for this 1 hour presentation. The other tools that I'm going to show, I'm going to gloss over more quickly out of necessity for the time, but I think the focus on two-level design and the process of analyzing that carries forward very nicely then into the more advanced tools. By the way, I just wanted to mention that back in our seventh Euro conference in Paris, where I attended and was one of the moderators, Dr. Daniel Trey, who's at the time was in part of the API chemistry group at GlaxoSmithKline in England, came and he presented a case study showing how they use DOE in their early phase development to rapidly screen reagents and solvents in a structured manner. And if you're interested in this talk, it's posted at this link, and you can find this, via that webinar page that I pointed you to before. Go to the main Statis website, go to webinars, find this presentation, and then if you download the PDF of the presentation, you'll be able to use the link. And take a look at that if you're interested. This happens to be a resolution 4 design, 2-level factorial, 5 factors, 20 runs. So this design, if we were to build it, 5 factors. Okay, so we're starting here. 2 blocks. Now notice that when we go to 2 blocks, the color turns yellow. So we lose some 2-factor interactions in the process, and the program updates the resolution to show that. 2 center points per block. And that should be 2 center points per block that 4 runs. So let's just see I don't know what the name of the factors are, but we could easily build this up. And this is a nice example of a block design where you can't maybe run it all in one day. And so you break them up into 2 groups. So here's a 20 run design. And these are just listed in coded values. The center points are listed with the zeros, indicating that the mid-range between -1 and +1. And if we were to type in names and levels, that would propagate through with an actual design. So go back and take a look at that if you're interested. But I thought that was pretty cool to see, testimonial from a company as big as GSK, which is our biggest licensee, I believe. Now let's just talk real briefly on the optimization side of things, where we're going to use response surface methods. This would be done only after you work through the screening and characterization, and you see the need to do response surface methods because of nonlinear behavior, with significant curvature. In this case, we've been able to focus our attention, hopefully, on a region of interest that would fall within a box like this, because then we could approximate the curve with simply a quadratic polynomial that has, squared terms in it. And this was exactly the sort of thing that I working on natural vitamin E. I had a chemist, a PhD, Bill, who only knew OFAD. And so he had a pretty good idea from 1 factor at a time that if he set A at a certain level, and then if he tested B, he could get that level of it. And so I said, Bill, I'm going to redo your experiment with a full 3-level experiment, which is a form of a response surface methods. And I was able to then develop a model that was a function not only of the main effects and the interaction effect, but also I was able to add these squared terms. And this forms what's called a quadratic polynomial. From that, using Design Expert, I was able to show that we could get the yield up from about 77% up to 95%. That was worth $2 million a year. that yield improvement for the natural vitamin E. So I can tell you that this multi-factor approach is definitely superior. And of course, RSM is another example of multi-factor. The only difference is we're using multiple levels. So in this case, after we go through factorial design and we really can narrow down on a few factors, we then can apply it to our process and then fit this polynomial model. This technique of RSM was invented by George Box. And in 1951, he came up with a structure called a central composite design. It's a design that not only has the two-level design with center points, but it adds further levels outside of the original square. And this central composite design is kind of a key design that we use for responsiveness methods. But what we have to keep in mind is that it's empirical. And so as George Box said, all models are wrong, but some are useful. If we can come up with useful models that predict well within our space, then that'll get the job done to move to a better place. So here's an example of fermentation optimization. You can see the structure of central composite design now with three factors. And they just looked at two different inputs, and they had their main goal to make this antibiotic, but they also wanted these other attributes to be maximized on biomass, oxygen uptake, and glucose. Here's the design that has the square part of it, the star part of it, and then the points at the center, listed in standard order. And here's the end result that they got, which was a nice little sweet spot where they could meet all the requirements. And this design is built up using Design Expert or Statis 360. Under the responsiveness part, central composite, there's various options which I won't get into, but this follows, the structure that George Box would have recommended to make a rotatable design that gives you equally precise predictions, equally distant from the center. We get a layout and a randomized plan, and so then basically the pharmaceutical chemists can just put together these combinations of things and then measure the results. Enter them in or copy and paste them in, and then we can fit these models to it, which incorporate the squared terms. After this, it's pretty similar in terms of looking for p-values overall, making sure that you have significant terms. In this case, we've got some terms that are insignificant that are supporting higher level terms, but possibly when you get into responsiveness methods, you might consider reducing out significant terms subject to model hierarchy. And so for in this model, I was able to reduce out those two-factor interactions and give, a simpler model. We still have to have B to support the B squared. And with these models, we end up with these really cool looking contour graphs oftentimes. In this case, you can see the drug, production is going to be maximized at the middle of the glucose and PFC. And then basically we do the same thing with all the other responses. Now, in the end, we have a series of models, which I can show on this coefficients tables. So each of these attributes has got a model associated with it. Some have got all the terms and some are reduced like I showed you. And then we just have to set up our goals. We want to maximize the drug production, biomass, oxygen uptake, glucose uptake. Program does a search for us, and here's a solution that gives us at least, some of what we want above the minimum thresholds for each of these attributes. Now, if we wanted to, I would assume that the antibiotic is going to be the most important thing. We can assign a relative importance that's higher for that. And that would bias the results to give us a little bit more antibiotic. Okay, now the overlay plot shading out what doesn't meet specification. And we can see that there's various boundaries here that are shown when I click, what's bounding this surface biomass. Now I have that one. When I click this, antibiotic turns red, and so we get some idea of what the limiting, responses or attributes are for our sweet spot or our quality by design. Now, I wouldn't really call this quality by design until you put in the confidence interval. And this one wasn't really sized large enough properly to get that confidence interval in. You need to have more runs. Let's see if there is one. Okay, there's a little sweet spot right in here that popped up. So it looks like it is possible to hit a sweet spot on this somewhere in the middle here. So if I was to reopen this file, I could show you that. So right in here with the confidence intervals is the sweet spot if we converge in on that. And so this would be the quality by design if we had that confidence interval. And here's just another example, but the time is too short for me to demo it. But this happens to be another pharmaceutical example. They just said they had parameter 1 and 2 for confidentiality purposes, but you can see the confidence interval has been put in here with this gray shading. And so this would be a QBD design space. Okay, the last part of it, which I may or may not show, depending on if there's any interest in the last 5 minutes or so, maybe 7 minutes, would be mixture design, where we're working to optimize a formulation. Before I talk about that, anybody who's interested in mixture design, would you raise your hands? Okay, Alan, Ken, Eva, and there's at least 7 more that are listing down here. So let me just give you, Mark, Eric, a quick view of this. Now, I actually like mixture design as much as anything because of it being in a triangular graph, which I think is really cool. And I learned this in material science that when you're developing something like a stainless steel, if you use these triangular graphs that go from 0 to 100% from the bottom to the top for ingredient A, and they go from 0 to 100% from right to left for ingredient B, and 0 to 100% from left to right for ingredient C. The nice thing about this is once you know any 2 ingredients, the third is fixed. And this really works well when you have a fixed total, which is typically 100 weight percent is what I suggest is the easiest thing. You have a formulation, it could be a subset of the formulation. For example, we have a tutorial where the total is kept at 9%. For the experiment, that 9% is varied, 91% is held constant. So that can work as well. And I won't get into the gory details of what happens with what I call the new hammer syndrome, when people get convinced that 2-level design is the way to go. But suffice it to say that this smug graduate from our MDOE class, our modern DOE class, thought for sure he could get the jump on the kids in the neighborhood to make lemonade by doing a little 2-level design. And he ended up doing an experiment that didn't really make any sense, because one of the conditions was 1 lemon to 1 sugar glass, and one condition was 2 lemons to 2 sugar glasses. So that didn't really make any sense at all. So what we want to do is recognize that as we start off with a pure lemon squeezed into into 1 glass, but then we start to add sugar water to it, the lemon juice has to be reduced to maintain just 1 glass of water. And the bottom line of this particular experiment, if you do it properly, by squeezing out a lemon, trying some pure sugar water, doing different blends, is you're able to see a synergism between the 2 of them that's very valuable. And this would be modeled by a specialized model that was invented by Henri Chaffee. It looks a little bit different, but we have this nonlinear blending term that is very informative, that if it's positive, like it is in this case, that there's a synergism. If it was negative, it would indicate a curve more like this. So I find that mixture design, and I would highly encourage you to study this, is very valuable. For example, in metallurgy, we can see that if we go 18% up on the chromium axis, and then we go 8% on the nickel axis, that once we do those two lines, there's an intersection, we can just read off the graph that there has to be 74% of the iron. The total would be 100 weight percent. So once you know any two ingredients, the third is fixed. And what I may need to do, because the time is going to run, is come back and do a part 2 of this presentation and focus on just the mixture part of it to really do it justice. So what I would suggest is, for those of you that are interested in mixture design, if you're interested enough, send me to mark@staties.com a request, for a follow-up mixture uh, presentation on pharmaceuticals only. And then I can do, a better job of spending the time that's necessary, because I got a little bit carried away with the two level and, the response surface methods. But just to give you an idea on the prize of doing two level design, this is how easy it can be with Design Expert or Statis 360. Well, basically, you're going to go down to this mixture part. You're going to enter in your ingredients. In this case, we're just going to leave it at 0 to 1 scale, which is equivalent to 0 to 100%. And I think these are different surfactants that were used in making a pharma prep, which was a film. And it ended up that they were focusing on particle size and the glass transition. And you get this recipe sheet. This is what I love about the software, is you get this recipe sheet. So basically, as a formulation chemist, you're putting these together, you're collecting up the data. And then basically, you're fitting a specialized model, which is a form of quadratic, but specialized by Chaffee, perhaps reducing it down. And you're able to develop these really cool graphs that show you what the response is throughout the region of the triangle. And you can do that for all your responses. Then you can do your multiple response optimization. Let's say minimizing R1, minimizing R2, and the program dials up a solution. And then you can overlay the plot. And even in this case, putting in the confidence interval, so now we have a quality by design. So that's a demonstration of how it works, and it basically builds off of response surface methods, but within the triangular space rather than the square space. And there's your sweet spot framed by the confidence intervals. That basically concludes my presentation. I just want to say, for reference sake, you may want to pick up one or more of the series of books that I'm the prime author of that do keep it simple and make it fun, not like the standard statistical textbooks. And it's focused in on two-level designs in the first book, and then designs that have more than 3 levels, which is response surface methods, in the second book. And then for the third book, it focuses on the mixture design, which is the triangular spaced experiments. And that's readily available from Amazon or the like. And then if you'd like to sharpen up your skills, we have public presentations of modern DOE, which is for process type optimization. We have a class that focuses on formulation development, which is called mixture design. And then for private presentation only, contact our workshops team. We have a specialized class that focuses on all of the tools for pharma only. All right, well, I hope that I succeeded in meeting my goal of a WIFM, of what's in it for me, of learning how these multi-component and multi-factor approaches are going to be the way to go to quickly, maybe 50% less development time or more, converge on a quality by design spot, and probably something a lot better than you could ever discover just doing one factor at a time. So I really think this is something that you did well by attending this briefing, and should continue to learn about these tools if you're not all the way up to an expert level. So I would say that whether you're new or experienced at DOE, that this talk was for you and your organization's bottom line. But that's easy for me to say. Do you agree? All right, I'm getting some positive feedback. I don't have a list of the entire group, but I see a very goodly number of people that feel that this was a worthwhile presentation. So thank you for that positivity. But anyways, thanks again for attending, and please do get back to me with any questions to mark@staties.com, and then I'd be very happy to link up with you if you want to follow this link for my LinkedIn at markstat. All right, so take care.